Today's lecture is really the first that is going to dive into our understanding of DNA and gene expression, as is the topic of molecular genetics. So what we need to do is really take a few steps back and make sure that we all have a good, solid understanding of the foundation of DNA structure. So before we do that, we should always start off with understanding some basic definitions. What we will focus on for the next few lectures are nucleic acids as a whole. Nucleic acids fall into two broad categories. There is DNA, which is short for deoxyribonucleic acid, and RNA, which is short for ribonucleic acid. And again, the two of those are the two main categories of nucleic acids. When we talk about nucleic acids, we know that they fall into the broad scheme of things known as macromolecules. So if you go back to some of your basic introductory biology, you should recall that there are four main macromolecule categories. There are carbohydrates, proteins, nucleic acids, and lipids. Now, three out of those four, proteins, carbohydrates, and nucleic acids, are very similar in structure. When we talk about macromolecules, again, those three categories that I just identified for you, we know that they are polymers, which means that they are long stretches or strands of repeating units over and over and over again. Those repeating units are known as monomers, or the monomeric unit, and those can be identical units or very similar units that repeat in a polymer. The typical analogy I usually give in my introductory classes is that a polymer is like a strand of pearls, where each pearl represents a monomer or a monomeric unit, and the strand of pearls is a linkage of those pearls, one right after the other, to give the polymer structure. So when we talk about polymers, that's the overall scheme that we are looking at in general. Now, since we're focusing on nucleic acids, what we want to do is look at that monomeric unit in a lot more detail. The individual unit or monomeric unit is very loosely referred to as a nucleotide. In a few slides, I will talk about the specifics associated with the definition of a nucleotide and modifications to that as it applies to molecular genetics. But for now, we're going to loosely refer to that individual repeating monomeric unit as a nucleotide. Every single nucleotide has three main components to it. The first component is a 5-carbon sugar, also referred to as a pentose sugar. There are two different types of pentose sugars that we can find in nucleotide structure. There is, on the left-hand side, ribose, and on the right-hand side, deoxyribose. If you look at the structures, they are almost identical. What I do want to highlight is that, again, they are 5-carbon sugars, and you can see the carbons numbered with a 1, 2, 3, 4, and 5 working in a clockwise fashion if you look at the structure overall. That numbering system is exactly the same in ribose as it is in deoxyribose. They do have the little prime designation after the numbers, so it's actually 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. And that's to differentiate the carbons from an additional component in nucleotide structure, which is known as the nitrogenous base, and we'll get to that in just a minute. But again, what I want to highlight is that these molecules are almost identical, and the only difference is at the 2' carbon end, which is what you see in ribose is a hydroxyl group, and in deoxyribose, the oxygen is missing, hence deoxy, missing the oxygen. That's the only difference between these two structures. Ribose is the type of sugar that is found in RNA nucleotides, and deoxyribose is the type of sugar found in DNA nucleotides. And as you can see on this slide, the proper designation for deoxyribose is actually 2-deoxyribose because that indicates the carbon where the oxygen is actually missing. Now, as I mentioned on the previous slide, there are 3 components found in every single nucleotide. The 5-carbon sugar, which differs between an RNA nucleotide and a DNA nucleotide, which we saw on the last slide. The next component that every single nucleotide contains is a phosphate group. If you look at the figure that you see here on the slide, what you're looking at is what we would refer to as the phosphate group as a functional group, where the R indicates the rest of a molecule where the phosphate group can be added. This is the structural characteristic or feature of a typical inorganic phosphate. However, we know that the hydrogens actually can be released rather easily depending upon the pH of the solution. And at physiological pH, 2 hydrogens do actually get donated into the solution, or 2 protons, as you can see in this particular figure. So again, at physiological pH, the structure of the phosphate group looks more like the figure on the right-hand side rather than the left-hand side. So when we talk about phosphate groups in general, the left-hand side figure is what we would typically draw, but at physiological pH, what we would see if we could look at the molecular structure is what you see on the right-hand side of this figure. Now, every single nucleotide will contain at least 1 phosphate group, but could contain 2 or 3, and again, I will review this on another slide. And I also want to add with this particular figure and slide is that when we talk about nucleic acids, the term acid is in the title. The reason that DNA and RNA are considered acids is that physiological pH, they do in fact donate or release that hydrogen ion, in this case 2 hydrogen ions per phosphate group, to the solution. Hence, they act as an acid at physiological pH. And again, the name deoxyribonucleic acid or ribonucleic acid because of this particular characteristic or feature that we just talked about. And lastly, for every single nucleotide, there is a component known as a nitrogenous base. It is a nitrogenous base because it has a significant amount of nitrogen in it. There are 2 broad categories of nitrogenous bases that can be found in nucleotides. There is what's on the far left-hand side, a generic form of what's known as a pyrimidine, and then below that is what you see as the generic form of a purine. When we talk about pyrimidines, it is a 6-membered ring structure, and there are 3 different types of pyrimidines that exist: cytosine, uracil, and thymine. I won't ask you for the exact molecular structure of any one of the individual nitrogenous bases, but you should definitely know the difference between the 5-membered ring, which represents the pyrimidine, and then the figure that's below that, which is, the pyrimidine is a 6-membered ring, not a 5-membered ring. 6-membered ring for pyrimidine. The figure that's below that is the purine, which is a 5-membered ring fused to a 6-membered ring. And for purines, there are 2 distinct types of purines, guanine and adenine. So again, I won't ask you for specific structural detail for any of the individual nitrogenous bases, but you absolutely should know that cytosine, uracil, and thymine are pyrimidines and are 6-membered ring structures, and guanine and adenine are purines, which are more complex overall, 6-membered ring fused to a 5-membered ring. Okay, so this particular figure or slide is the one I was alluding to just a few minutes ago with one of our previous slides. And that is, this gives us a little more detail and specific definition associated with nucleotides. So again, a nucleotide is a very generic term that is a molecule that contains the 5-carbon sugar, a nitrogenous base, and at least 1 phosphate group. But nucleotides can contain 2 phosphate groups or 3 phosphate groups. So the nucleotides are actually a family of molecules that have distinction associated with them with the number of phosphate groups that are added onto the structure. If you look at the first term on the slide, underneath the bold heading, we see the term nucleoside. And the term nucleoside defines a base bound to a sugar with no phosphate group. The nucleotide then is going to be the base, the sugar, and at least one phosphate group. What you see on the far left is an example of a monophosphate nucleoside. So the proper terminology for this figure is, this happens to be deoxyadenosine. So the base is adenine. The correct terminology here is deoxyadenosine monophosphate. In the figure in the center, you have deoxyadenosine diphosphate, and then the one all the way to the right, deoxyadenosine triphosphate. So again, these are all considered nucleotides, and when we use the word nucleotide, we tend to use it very loosely. However, there are specifics in terminology and definition of molecular structure, and you do need to be aware of those differences, as they're not necessarily interchangeable. And we'll get to why that is when we start talking about DNA structure, but more specifically replication and transcription. The differences in structures are very important. Now, what I want to highlight with this particular slide is some very important features and characteristics associated with nucleic acids. And that is, nucleic acids are in fact polymers. That is, they are linkages of nucleotide after nucleotide after nucleotide in a long stretch, which is again the defining characteristic or feature of a polymer. And with most polymers, there is a directionality or polarity associated with the molecule. If you look at the figure as it's drawn on the slide, you see 3 nucleotides that are linked one right after the other. So this technically is a trimer, but if we added more nucleotides onto it, it would eventually be a polymer. But there are 3 nucleotides that are covalently linked one right after the other. What I want to highlight, though, again with this figure is the directionality or the polarity of the molecule. And that is, in the top of this figure, you see the phosphate group, the sugar, and the base of a given nucleotide. Then in the center, you can also follow along and see the phosphate, the sugar, and the base. And then down at the bottom, again, the phosphate, sugar, and base as the building blocks to build this polynucleotide. Back at the top of the figure, you can see that the phosphate group is highlighted in blue. Phosphate group is highlighted in blue at the top of the figure. And this phosphate group represents one end of the molecule. This is known as the 5' end, and that's because the phosphate group is linked to the 5' carbon of the sugar, and it is not covalently bound to another nucleotide. It is essentially free. So the proper terminology is the free 5' end, but we oftentimes just shorten it to the 5' end. If you look at the bottom of the figure, you see again in blue a 3' hydroxyl group highlighted for the bottom nucleotide. Again, we're focusing on the sugar itself, and that is the number 3 carbon that the hydroxyl group is highlighted on. But again, for sugars, we have the prime designation for the carbon, 1', 2', 3'. So that's the 3' carbon. And what you see is that the hydroxyl group is linked to the 3' carbon, but there's no additional nucleotide covalently bound at this position, hence it is free. If you look at the center nucleotide, the phosphate group is highlighted in pink, and you can see that the phosphate group is linked to the 5' carbon, but also covalently linked to the nucleotide above it. If you look at the 3' carbon of that same central nucleotide, the 3' carbon is covalently bound to the phosphate group of the next nucleotide. So there's no free 5' end or 3' end for that very central nucleotide in this figure. So you should be familiar with looking at basic structure like we have here and identifying where the 5' end is and the 3' end. Now, oftentimes by convention, we tend to put the 5' end at the top and the 3' end at the bottom, but that's not necessarily always the case. When we talk about the double helix of DNA, which we're going to get to in just a minute, the two strands are going in opposite directions of one another. So one strand is essentially right side up with the 5' end at the top and the 3' end at the bottom, and the other one is comparatively upside down, 3' end at the top, 5' end at the bottom, and we'll see a figure that highlights this in a little more detail. But again, you should be familiar with nucleotide and nucleic acid structure, where you should be able to identify 5' and 3' ends of a polynucleotide. All right, so now we're going to talk, now that we've understood the basic nucleotide structure, that building block that's required to build a nucleic acid, what I want to talk a little bit more about is the structure of DNA overall, and tangentially just a little bit the structure of RNA. So hopefully what you are all familiar with is that Watson and Crick were the ones who are credited with discovering the structure of DNA. And at this point, you hopefully are all familiar with the fact that the DNA is in fact a double-stranded molecule that is in a helical shape. But Watson and Crick were the ones who are credited with our understanding of what that shape and structure really is. I always like this particular figure because the picture on the left is sort of like their promo shot that they took for all the press that was going on at the time, where they were standing in front of the model. And I always thought it was kind of funny that they, stood in front of this model for this promo type of shot. But the reality is that they were actually working with molecular models, kind of like what you would do in a chemistry class, where you would put atoms together and build molecules and look at complexity associated with that. And they were actually doing that as part of their work. So it wasn't that they put the model together just to make a picture. They were actually making the model to try and determine the structure of DNA overall. The figure on the right is a really nice one that was done at the 50th anniversary of the discovery of the structure of DNA, and they sort of just reenacted that classic photo, and I just thought that one was kind of interesting. All right, so what I want to talk a little bit about is how Watson and Crick came to their conclusions overall, in that they actually did not work alone in coming up with the idea that DNA was in this double helical shape in its natural state in the cell. So what they did know came from the work of a scientist known as Rosalind Franklin. Rosalind Franklin was a female scientist that worked in the lab of Maurice Wilkins, who actually had a lab right next door to Watson and Crick. Now, at the time, women weren't necessarily given the credit that they were due. She did in fact have a PhD, so it wasn't like she didn't have the know-how or the knowledge. But because she was a woman, she had a really hard time establishing her own research lab, and so she worked in the lab of Maurice Wilkins. Now, what she was able to do was perfect this technique known as X-ray crystallography. Very simply put, the idea is to make a crystal structure of the molecule that you're interested in understanding or learning the structure of. And you essentially take that crystal structure and expose it to X-rays. There's a very complex mathematical calculation that goes along with it, and then essentially you're looking at the diffraction pattern, how the X-rays are scattered when they are shining on the crystal structure of the molecule in question. And this particular figure that you see on the left-hand side is the classic figure that helped establish that the DNA structure was in fact a double helix. But this particular figure was the one that suggested the helical shape of the structure overall. As the story goes, it turns out that Rosalind Franklin didn't actually just give these figures to Watson and Crick, but Maurice Wilkins handed them over to them sort of in a pub conversation over a couple beers. And so she kind of was, her data and her work was taken from her unknowingly. And unfortunately, she passed away before the Nobel Prize was awarded, and they do not award the Nobel Prize posthumously, meaning after someone has passed. Okay, so they had that bit of evidence, the figures or the images that Rosalind Franklin was able to generate that actually suggested there was some type of helical shape associated with DNA. They also had work from electron microscopy. Now, the work that they had from electron microscopy gave them some idea of the size of the molecule overall. They knew that the helix, or the structure of DNA, before they knew it was a helix, was 20 angstroms wide. Now, angstroms is just a unit of measure associated with very, very small objects. Just to give you some idea, the thickness of a piece of paper is 1 million angstroms. So we're talking about something that is very, very small if we're talking about 20 angstroms overall. But electron microscopy data was able to demonstrate that the width of a DNA molecule was 20 angstroms wide. And working with their models, like we have again in this figure on the right-hand side, is that they were able to establish that nucleotides, if you had two purines sort of paired up with one another, two nucleotides that each had a purine, that would be too small. And if you had two pyrimidines that were paired up, one after the other in nucleotide structure, that would be too big. And if you had a nucleotide with a purine paired up with a nucleotide with a pyrimidine, that would be right about the right size. So again, this gave them some indication of the structure overall. Watson and Crick also had the data that was collected by Erwin Charga in the early 1950s. Erwin Charga was a biochemist, and one of the many things that he did was isolate DNA from many different types of organisms. He was able then to hydrolyze the DNA, which basically means just break it up into individual nucleotides. He could then perform a simple chromatography to separate out those nucleotides, and he could quantify the nucleotides in a given sample. What he found, and we'll talk about some of this I think in our next lecture, was that there were some really interesting comparisons that could be made when discussing nucleotide composition. One of the things that came about was that organisms differed in their nucleotide composition. So, for example, one organism may have had 25% A's, but another organism may have 35% A's. So there was variability in the concentration of given nucleotides from organism to organism. But the most important thing that came about from his work was what's referred to as Chargaff's rules and is also referred to as sequence complementarity. What Chargaff was able to do with his analysis was determine that no matter what species he isolated DNA from, the concentration of A always equaled the concentration of T, and the concentration of C always equaled the concentration of G. And again, that relationship did not change from species to species. And that was a really interesting concept. And what Watson and Crick did was take that one step further and refer to this as sequence complementarity. This goes back to what I just mentioned on the last slide, was that with the 20 angstroms wide DNA structure, you could have a nucleotide with a purine paired up with one with a pyrimidine, and that would be the right size or the width. And what you see here is that base pairing rule. A's will always hydrogen bond with T's, and G's will always hydrogen bond with C's. So Watson and Crick were able to put all of those pieces of the puzzle together to come up with our current conceptual understanding of DNA in the cell. We know that the structure of DNA, as it is found in its physiological state, is actually two separate polynucleotide strands that come together to form a double helix. Double helix because it's two strands that make up the helical shape overall. What you see on the left-hand side is the classic structural characteristics of the double helix. If you unwound the helix, basically held one end in your hand and twisted the other end so it unwound, you would have a structure that looks very much like a ladder, and we'll see that in the next slide in just a minute. But what I wanted to highlight are just some of the structural characteristics or features for DNA. Again, it is a double helical molecule. It is a right-handed helix, which means that the helix turns. If you take your right hand in towards your body and turn the spiral upward, that's a right-handed helix versus a left-handed helix, which would be going in the opposite orientation. The bases fit together via hydrogen bonding, and what's highlighted in the pink is the sugar phosphate backbone portion of each of the polynucleotides. Now this figure is one that I tend to show in just about every one of my classes, all the way to my introductory to this more advanced molecular genetics class, and that's because it shows you different views overall of DNA structure. So the one in panel A is exactly what we were just looking at on the last slide. In this case, you've got that tealish blue color is the sugar phosphate backbone of each of the polynucleotide strands, and then you can see the bases pointing in towards one another, hydrogen bonding, and it is that hydrogen bonding that holds the two strands together. If you unwind the helix, like I mentioned in the last slide, you get a structure that looks a lot more like a ladder, and you can see the sugar phosphate backbone on either side, which would be the handles that you would hold on to as you're climbing up the ladder, and then the rungs where you would put your feet is where the bases actually are found. Now, DNA, it consists of two polynucleotides, and they are anti-parallel, which means they are running in opposite direction of one another, like I mentioned in one of the previous slides. So in panel B, you can see the two strands coming together. The left-hand strand in panel B has the 5' end at the top and the 3' end at the bottom, whereas the right-hand strand has the 5' end at the bottom and the 3' end at the top. And I really like this figure because it clearly shows that upside down orientation of the right-hand strand compared to the left-hand strand. And then you also have a space-filling model just to give you some idea of the structural characteristics of DNA. Now, when we talk about RNA, RNA is typically a single-stranded molecule. It's not like this complex double helix. However, there is a lot of what's referred to as secondary structure for RNA, where hydrogen bonds can form between adjacent or nearby nucleotides, and that does give secondary structure. But they are truly tend to be single-stranded molecules that may fold up on themselves, but not two strands coming together like you see for the DNA structure. Early X-ray diffraction studies indicated that there were in fact 3 different forms or structures of DNA, referred to as B, A, and Z. Now, we'll talk about B first, and B is what we would refer to as sort of being the ideal structure. That's the one on the far right-hand side. This is the structure that's observed at high humidity. It is the average structure that you would see under physiological conditions. Just to give you some idea, there are about 10 nucleotides per turn of the helix. The A form, which is the one you see on the far left-hand side, looks kind of squished in comparison to the B form. This is observed at low humidity. Oftentimes the structural characteristics that you would see when you pull DNA away from cellular conditions, so not physiological conditions. It's a bit more compact in that it's about 10.7 nucleotides per turn, and again, it looks a little bit more squished overall than the B form. But again, the B form is what we would typically find at physiological conditions. And then lastly, in the center, you have what's known as the Z form. And the Z form is a left-handed helix, which again, I highlighted just a minute ago that our physiological form is a right-handed helix. It has a conformation change along with a little bit different linkages that can be found between the sugars and the bases. This is a little bit more stretched out form in that there are 12 nucleotides per turn. It's found in association with high concentrations of positively charged ions like sodium. It is not stable. It's considered transient. And initially, it was thought that this particular form was found sort of by mistake, in that it was an artifact of extraction procedures. Well, there is more recent research that suggests that it could be important for transcription, and when we get to transcription, we'll come back to this particular form and talk about it in a little more detail. So what I want to do now is transfer over a little bit and talk not so much about the structure, the physical structure like we just did of DNA, but talk about some additional characteristics that are associated with DNA just to get a feel for variations that we know occur. So the first thing that I mentioned with Charga was Charga's work was able to demonstrate that there were differences in concentrations of a given nucleotide from organism to organism or species to species. And oftentimes what is utilized to differentiate this is to refer to the GC content, G plus C, and its percentages. So what you see in this particular table, it's a little bit dated, but it's a really nice table that shows you quite a variety associated with G plus C content. So at a very low percentage, at 22%, you have something known as dictyostelium, which is a type of mold or fungus. And as we move down the table, eventually you'll see you get to calf thymus, which is DNA isolated from the thymus of a cow or a baby cow, rats, bulls, wheat, and the concentration or the percentages actually go up. And all the way on the right-hand side, at the bottom of that table, you can see something with a very high concentration of the G's and C's. And so this just shows you the variability that occurs from species to species. And it's not one size fits all for concentrations of individual nucleotides. What I do want to highlight is that when we talk about complementarity, we know that it's hydrogen bonds that hold the bases of the nucleotides together. A's will always hydrogen bond with T's, and G's will always hydrogen bond with C's. So when you have A's and T's together, there are 2 hydrogen bonds that can form between A's and T's, whereas 3 hydrogen bonds can form between G's and C's. So a DNA from an organism that is very GC rich is going to have its DNA held together much more tightly because there's more G's and C's, more hydrogen bonds that can form, because 3 can form between G's and C's compared to A's and T's. So that's just something to keep in mind, again, about the differences in behavior of DNA when we talk about that GC content overall. And this is actually important with our previous lecture discussing PCR. And with PCR, we have to have the denaturation of the DNA. We also have to have the annealing of the primers, and that temperature varied from primer to primer. That's because of the GC content overall. So this slide relates to that concept that I was just talking about with the amount of G's and C's that you find in a double-stranded DNA molecule will play a role in how tight those two strands are held together because of 3 hydrogen bonds versus 2 for every GC pair versus 2 for every AT pair. So that leads to then this topic of what's known as the melting temperature of DNA. So one of the things that we just talked about was for PCR, if you take DNA to almost boiling point, that weakens those hydrogen bonds enough to denature or pull apart the two strands. Now that particular characteristic can be utilized in analyzing DNA in what's known again as the melting temperature. So before we get to that, what I want to address is that double-stranded DNA absorbs light at a wavelength of 260 nanometers. The double-stranded DNA, the two strands quench the absorbance. And what happens when you have single-stranded DNA is the absorbance actually rises by about 30 to 40%. So you can actually determine when DNA starts to go from a double-stranded form to a single-stranded form by this change in the absorption at 260 nanometers. What you can also determine is what's known as the melting temperature. And the melting temperature, designated T with the subscript M, is the temperature at which DNA strands are half denatured. So this is, again, some indication of the content or the composition, because a higher melting temperature is going to indicate that it's going to take more energy to pull apart those strands, which means most likely there's more G's and C's than A's and T's. So this is my own table that I've put together just to make some comparisons from species to species. I have some prokaryotes and I have some eukaryotes on this particular slide. What I want to highlight are some additional definitions, and that is of a chromosome. So a chromosome is a physical structure composed of DNA and proteins. Now, the number of chromosomes varies from species to species, and that's just, again, a number of physical structures that are utilized to make up the genomic information of the organism in question. So what you see in the first column is the number of chromosomes that you can find in these organisms. And what I really like to highlight is that in eukaryotes, you have something that is a single-celled eukaryotic organism, Saccharomyces cerevisiae, which is baker's yeast, versus Homo sapiens, which is humans. And you can see that the number of chromosomes that make up the genomic content is not that different. And then if you look at something like Arabidopsis, which is a model plant system, there's 5 chromosomes, and Drosophila, which is the common fruit fly, made up of 4. And so the number of chromosomes really doesn't mean anything other than that's the number of partitions that are utilized to make up the genome of that particular organism. In addition, in the next column, you can see some characteristics as far as the structure of the chromosome. For prokaryotes, most prokaryotes have a circular chromosome, whereas most eukaryotes have linear structure associated with their chromosomes. And then in the last column, what you can see is this great variation in genome size, which just means how many bases make up the genome of a given organism. So you have 2 prokaryotic examples on the slide, and those genomes are pretty similar in size. And then you look at eukaryotes, and you can see that as you increase in complexity of the organism, comparing a single-celled eukaryotic organism, the Saccharomyces cerevisiae, to Homo sapiens, there's a dramatic jump in the genome size. So what you see in this particular table is some modifications associated with that data. and that you have the genome size repeated again in the first column. What I've added in is the approximate number of genes, and those numbers change regularly, and so the numbers on this slide might not be exactly what the numbers are today, but that's really not important. What you can see is just a comparison overall in the approximate number of genes. But what I really want to highlight is what's referred to as the gene density. So the gene density just refers to the average number of genes that you find per megabase pair of DNA. So the gene density in something like a prokaryote it's very dense. The genes are like one right after the other. As you increase with genome size, we're actually seeing the genes be much further spaced out in something like Homo sapiens, for instance. So the question is, why do we see this difference in gene density? So the first answer is the simple difference between a prokaryote and a eukaryote. But if we then look within eukaryotes, there's quite a bit of variation associated with that as well. So there are increases in gene size that can occur. This includes things like promoters, regulatory sequences. These are sequences that direct and regulate transcription. There's much more complex mechanisms associated with regulating transcription, hence that might account for some of that difference. There is an increase in DNA between genes, which is referred to as intergenic sequences. And we're going to talk about that in just a minute. But we also know that for eukaryotes, protein coding genes have interspersed regions of non-protein coding regions, which are referred to as introns, and then protein coding regions, which are referred to as exons. And hence, that also does account for some of this difference. It turns out that approximately 5% of a gene in humans directly codes for protein. 95% of the gene does not. So let's talk a little bit about those intergenic sequences. Intergenic sequences are regions that are not associated with the expression of proteins or functional RNAs. And we're going to come back to this functional RNA concept later on in the semester. For example, greater than 60% of the human genome is intergenic, and most of it has no known or no currently known function. When we talk about intergenic sequences, there are two broad categories. There's what's referred to as a unique, which accounts for about 25% of intergenic sequences. These are non-functional mutated genes, oftentimes due to mistakes in DNA recombination. They could be gene fragments. They could be what we refer to as pseudogenes, which are the result of the enzyme known as reverse transcriptase, which is typically associated with viruses that copies RNA into DNA, and then that double-stranded DNA gets reintegrated into the genome. But they don't get expressed because they don't have regulatory sequences to allow for their expression. So, aside from unique DNA or intergenic sequences, there is the repetitive, which makes up about 75% of intergenic sequences. There are those things known as microsatellites. These are very short, typically less than 13 bases in length, and they are repeats. Most common is a dinucleotide repeat, and these are usually errors or due to errors in replication. In addition to microsatellites, there are genome-wide repeats, which can be greater than 100 base pairs. Many of them are greater than 1,000 base pairs in length. And this is just, again, another category of repetitive DNA. So we've got unique versus repetitive. And I think that's where we'll stop this lecture, and we will pick up with talking a little bit more about DNA organization in the cell in our next lecture.